Hello, my name is Ling Yang, and my oral presentation today will be on the Prader-Willi Syndrome, or PWS for short. So I just want to start off with a brief introduction on what Prader-Willi Syndrome is. It is an autosomal dominant disorder or genetic disorder that is described to be from the lack of expression on chromosome 15, specifically on Q11 to Q13 due to deletion of maternal uniparental disomy or imprinting defect. Now, this genetic disorder is rare, with a prevalence of 1 out of 10,000 to 25,000 births worldwide, affecting males and females as well as all ethnicities with equal frequency, and it is the most common form of obesity that is caused by genetics. Now, moving on to some signs and symptoms. These will vary between the stages of age of a PWS individual. Starting off with infants, you can see hypotonia, which is the weakness in muscle tone, feeding difficulties, poor growth, and delayed development. In later stages, in early childhood, those affected can develop extreme appetite, which leads to overeating and obesity. There are also some behavioral signs that one with PWS may have, which includes stubbornness, often having temper tantrums, and this obsessive compulsive tendencies, such as developing this hyperphagia or excessive hunger. So usually a PWS child after the age of 2 will start to have heightened focus on food and start to gain weight very easily, usually resulting in becoming obese. Now, the most common physical signs include having a narrow forehead. They often have almond-shaped eyes, usually crossed. They have a triangular mouth that's shaping downwards, the edges shaping downwards, and they usually have a very fair skin composition. And also, they usually have small hands and feet. As far as diagnosis goes with PWS individuals, you can see the symptoms list will vary and increase with age. So symptoms of hypotonia or the low muscle tone persists starting at birth to 6 years of age, and then there's a dramatic urge or need of having an increased appetite starting in those ages 6 years and older. As you move further with age, cognitive disabilities start to show around the preteen stages at ages 13 years old through adulthood. There will also be other symptoms in these age groups that include hypogonadism or typical behavior problems such as the temper tantrums and compulsive-like behaviors that we talked about in the previous slide. While there are many genes that are depleted in PWS patients, the ones I want to draw your attention to today are this class of SNORD115 or HB252, and these are actually going to be a cluster of genes that are small nucleolar RNAs or SNO RNAs. Now, I just want to remind you that SNO RNAs are a class of small RNA molecules that primarily guide chemical modifications of other RNAs, which may include ribosomal RNAs, transfer RNAs, and small nuclear RNAs. Now, this type of modifying RNA is usually located in the nucleolus of a cell, which is a major site of snRNA biogenesis, and it actually exhibits sequence complementarity towards this receptor, specifically the serotonin receptor 2C, also known as 5-HT2C. An experiment that was done by Kishore and his team in 2006, they have found that this SNORD115 regulates alternative splicing of the protein coding region of the serotonin receptor 2C. And they found that it does this by binding to a silencing element in the exon. So Prader-Willi syndrome patients actually do not express this protein. They have an mRNA isoform that's different from healthy individuals. So with that in mind, they concluded that a SNO RNA actually regulates the processing of an mRNA expressed from a gene located on a different chromosome, and the results indicated that a defect in this pre-mRNA processing contributes to the Prader-Willi syndrome. Now, although these cluster of genes is thought to not really play a major role in PWS, studies such as those done by Kishore involving microdeletions of these regions have shown to be associated with PWS symptoms such as syndromic form obesity and the hyperphagia found in these individuals. Now, the article that I looked at for my mouse model of Prader-Willi syndrome sought to see increased alternative splicing of the HTR2C serotonin receptor in these mouse models that had Prader-Willi syndrome, leading to the disruption of the serotonin receptor-mediated appetite. So they investigated the impact of an abnormal serotonin functioning in the mice with PWS, with a key aspect of focusing on cognition of these mice, and they found that abnormal post-transcriptional processing of the serotonin 2C receptor, the pre-RNA, contributes to aspects of the Prader-Willi syndrome behavioral phenotypes. Now, what they saw in these PW mice were that they were considerably larger than their wild-type litter mates. Symptoms that they showed often include increased appetite. They had aggression and also were often very fatigued. Other than the physical symptoms, they also had cognitive abnormalities, including enhanced appetite learning and reversal learning, and also deficits in visual spatial attention. Now, I do want to point your attention to the figure on the right, showing an increased levels of truncated serotonin receptor in those mice with PWS, looking in their hypothalamus region. The gene expression splice variant ratio that was assessed using quantitative PCR in a microdissected hypothalamus of these mice. And they found that expression of the SNORD115 was absent in those mice's hypothalamus with PWS, and this deficiency actually promotes alternative splicing of the serotonin receptor with levels that were truncated, often being non-functional, and were actually increased in PWS mice relative to those of their wild-type litter mates. So they concluded that there was a disruption in the mediated appetite and also led to these cognitive abnormalities that were seen. Now, some concluding thoughts and questions that I'd like for you guys to think about and kind of remember is that Prader-Willi syndrome is a complex disorder, and the link between genetic causes and the clinical picture as a whole is unfortunately only partially understood, which requires the need for more experiments to be conducted to further understand this rare complex genetic disorder. However, while there is no medical cure or prevention, early diagnosis of PWS may be essential. This will be able to give parents time to learn about and prepare for the challenges that may lie ahead, and to establish family routines that may better support their child's diet and behavioral needs just from the start. Now, from the experiment findings, they have shown a relevance towards appetite. So knowing this, PWS individuals may seek treatments or medications that control appetite. Just a matter of understanding and finding out what kind of treatments and medications may be available. And with that, these are my references. I want to thank everybody so much for watching. I hope you got something from my presentation, and thank you so much for watching.